In the machine shop? Take it now. Oh, it should be up. And then if we're up, then we should start. Now, even if we miss a little bit of window,
we have a different one if you want.
Cool. Yeah, we're a little late anyways. Thanks.
And if he wants a microphone, we can try and connect us again.
I think it's about time to sit down.
Hi, everybody, and welcome to the August meeting.
This is my chair. Oh, okay, yeah. That's your chair. My chair. He's right back there. Oh, yeah, there he is.
Thank you to everybody for coming out. We've got a lot in store tonight. We've got a very exciting talk. Alex Martin flew in all the way across the country to talk about Vallisneria. All you plant lovers are definitely in for a treat. And thank you. Oh yeah, of course. As always, we're going to start off real quick with a roll call, just for our record keeping. My name's Travis. Luke, our vice president, was just here. He'll pop back up somewhere. Modesta, our secretary, is in the back. We had a change. We've had some shuffling happen. And Tyler has received a promotion, has to step away to deal with his job, and Joey has graciously agreed to step in as our treasurer. So I don't see Joey in the room. Thank you. There's Joey. Thank you very much, Joey. And our Board of Governors, Jan Nolta, somewhere here. Thank you, Jan. Joe Murchison, over here at the auction table. Larry Schenkel, right over here. Thank you, Larry. Nico Greiners, helping check stuff in. And Matt Adams, I saw him here earlier. He'll be up here soon. Thank you, Matt. And then Ryan, who could not make it today. Thank you, Ryan. And we have a special announcement before we get started today from Randy's Fish Palace. If you want to take... Rand, do I see you?
I'll be right here.
Yes, if you want to come up and talk to the group, let everybody know what's happening. We have...
I can stand right here.
Yeah, wherever you're comfortable.
First of all, I'm so amazed that there is this kind of amount of people that's interested in tropical fish still. Over the years, we've been told about in the industry that the aquarium business is starting to become the way of the dodo bird. With regulations, thank God for your state of California that wants to, reduce things, and so the fun of it tends to be a more challenge when I get to see a group like this. I've been in business for about 52 years. I have had the privilege of working in fish farms and doing all the other things, and starting my own store. It said in 1976. And since then, I've run and operated quite a few things. And never would I have thought that the last day of my thinking that our store caught fire. So as a result, I am no longer in the business. Just because I'm 69 years old and feel that's a good way to end it in one way or another. Everything's okay, insurance, thank goodness, fighting them and stuff, they got taken care of. But in the meantime, I have a lot of items at the store, aquariums, from a 240-gallon custom aquarium that's made in back east, to cubes, iron-free stuff, 75s. I have some Mars units. Granted, these guys suffered a fire, but those aren't the ones that I need to get rid of, but would like to have help to haul them out so that I don't have to pay another $3,000 for the privilege of having my rent there. So anybody who has any thoughts to come down with Larry, I have to go back home to Oregon where I live. But Larry has graciously volunteered to help coordinate the idea of assistance, any of you come in to get the aquariums. I'm not in any way, shape, or form trying to make anything on it, other than having the idea of you coming and rescuing from there. The only thing I do not want is just people to grab something and run and leave the rest of it there. So I guess the only payments that I would ask is that you would assist in helping haul out a few items of each one of you. Some of the tanks are on racks. They're on large, those steel framed racks. Anyway.
Where is it?
It's in Stockton, California. That's just a hop, skip, and a jump. I'll pay for your gas by giving you tanks for free. Any other items that are there that can be salvaged, we can arrange it with Larry. And I want to appreciate any help that you can do. It's a time limit. It's not one of those things where I'll wait next week or what. It's now, and it's not like let's leave the club right now and go down there. But over the next several days, I've been requested to try to find homes for the things so the dumpster people don't come in and then they'll throw everything away. So it seems to me to be silly to have this kind of equipment with people here in this hobby who can utilize them, have foresight. Yes, they're not soot on them. Yes, it's going to take some elbow grease. Yes, you're going to have to wear rubber gloves and a mask. It is a fire issue, and some of them, but it just takes some elbow grease and some effort to benefit from that. So if you can help, please let the president of this club know, which he'll then let Larry know, and we can arrange it. So I'll be here until Wednesday. So tomorrow, Tuesday, Monday, Tuesday, Wednesday. So if you have any questions directed, yes.
Related, but did your big gut fish in the back, did you do okay?
He did not make it, but his girlfriend did. Someone rescued him and took him up to the foothills and put him, huh? Oh, yes. Thank you. So we did have someone rescued. There was a lot of fish that were rescued. Thanks to Larry, thanks to Joey, and other people who came because they would have not made it. Whoever said one day, how in the hell could a fire melt or start in a fish store? Well, it started with electrical fire from salt coming down into a circuit, which caused it to spark, and it caused the stand that it was sitting on to burn and emit that soot. And before you know it, all night long, my camera system caught it. From the time it started, I saw it, the ignition, and within 10 minutes later, everything was up, and the soot and the smoke did most of the damage. But luckily, there's a lot of things that's salvageable, but it just takes people with some vision to take that out of the equation is good. If you're really don't want to be bothered with little stuff like that, then it's probably not your as people say, your jam. But please let us know, because again, the landlords take over on Thursday. That is my story. Thank you for your moment.
Thank you. I'll definitely come help, and if anybody else wants to, just let us know. And do we have any other announcements? If not, I'm going to hand it over to Alex. Alex, thank you so much for coming to speak to us today.
Yeah, I'm Alex Martin. I guess there's no microphone that works, so I'll just try to shout. Right?
Real quick before we start, there's a large pepperoni pizza here without a number, I think. So anyone order that one?
Yeah.
Someone get that pizza.
Alex is hungry.
No, I ate before I came here, so I'm good. Alright, so if you can't hear me, just be like, hey, yell. Okay?
What? Yeah, what?
And I'll... Louder? Okay, this is gonna be tough.
No, you're good.
Alright, so if you have any questions while I'm giving this talk, just raise your hand and I'll try to see that you're trying to ask a question. If you don't understand something, we don't have to wait until the end. So, I recently just got my master's degree from the University of Kansas. I graduated last year in May. And so, thank you. So, I'm going to be talking about the project that I did to complete that master's degree. And so, before I get directly into it, I just want to give a quick background about it. Yeah?
Did you use this microphone?
It is not
It's not
working,
working.
unfortunately.
No.
The microphone is out of commission.
If you want, you can sit in the front and be melted in the fire.
So I just want to start off by talking about what aquatic plants are in general. So what makes a plant an aquatic plant? Well, it's a plant that lives all or most of its life history in or around water. And I've broken this down into about 4 categories. So the first one I'm going to talk about is the ones that grow in the shoreline or the riparian zone of like a river, the ones that you see right on the edge. And this includes things like Lobelia cardinalis. And it's really common in the hobby. We've had it for many, many years, especially people who Dutch aquascaping. And what people don't know that are just like traditional gardeners, like on land, is that it's been really common in the hobby here. And this is actually what it looks like when you're growing it like in the open air, like in a garden. It produces these really pretty red flowers. I actually just saw this in New Jersey where I was doing some work. And then the second type is emergent plants, so like Sagittaria, like this one, the arrowhead plant. This one was, I got this picture in Kansas, so when I was doing some research at the field station, this was growing in one of the ponds that we had. And emergent plants, they have their leaves and their flowers above water, but like the rest of it's kind of rooted in the water itself, like actual standing water. And then the third type is floating plants. I'm sure a lot of people are familiar with these. It's like duckweed, water lettuce, water hyacinth. I think water hyacinth is like federally regulated here in California, right? Like it's a big no-no.
Regulated to be everywhere.
Yeah, everywhere. And so these are just free floating. They move depending on where the wind and water move them to. And then the fourth and final type is submerged plants. So the topic of today's presentation is a completely submerged plant, Vallisneria. And so aside from the flowers that they produce, every part of the plant has to stay underwater in order for it to survive. But if you've taken biology, you know that there's some really weird stuff within biology in plants and animals. There's always exceptions to like a main set rule. There's one species of val that will grow out of the water. They have something called a mudflat form, and that's found in Australia. It's called Vallisneria erecta. And so why did I start studying aquatics? Well, like, why not when you can get an aquarium that looks like this, right? You can learn more about everything that looks like this. This is by far my favorite aquascape I've ever seen in my life. And I did get to meet Joe Harvey when I gave this talk at AGA in October at Chicago. Really cool guy. So I started growing aquatic plants in 2010, and I initially gravitated right towards the Dutch aquascapes. Like, it's the one thing that I wanted to do the most. There you go. And I've tried a few different times to mimic something like this. It is not easy. It is really difficult. And I learned quickly that Joe definitely has a special talent that he can get award-winning aquascapes like this. So, like in 2012, I got a 100-gallon rimless that I got custom-made from Miracles in Canada. And I tried a couple different aquascapes. This was like just throwing plants in the tank, see what grows in the water at my house. I tried some Dutch,
What is a Dutch aquascape?
A Dutch aquascape is focusing just on plants. There's actually no hardscape allowed in a Dutch. You have like certain streets of plants, so you have groupings of plants, and they're very limited to how many different species you can have. And you also want to have certain focal points. So they work a lot in like photography with thirds. So you have a focal point maybe in like the lower right-hand third, and then like another focal point up in the left other third of the tank. And it has to, like if you see the Joe Harvey tank, everything is grouped very neatly. And depending on the size of the tank, you can't have like too many species. You're very limited based on how many you can have with the size of the tank. And there has to be like a nice, aesthetic to it. They have, you can look up the rules. They have some very...
You're allowed one species of plant per 10 centimeters of tank length.
There you go.
Isn't it, I mean, I've talked to Joe, don't they only last a week or so before you have to take them down and reset because of the growth?
Here's the problem. Like, Lobelia cardinalis is going to have a different growth rate than this Vallisneria right here. So this val is going to send out runners. And so to get the tank to look perfect for this photo, he had to trim these plants and replant them all at different times because they all grow in different, one's fast growing, one's slow growing. So it's a lot of work, like every single day. So I gave a try at it. Figured out quickly it just becomes a jungle. Like there's like trying to get it to look nice. It's like it's like trying to hurt. No, it's like trying to hurt cats. It's really hard.
Jungles can look good.
Yeah. So when I became a grad student, they gave me an office on campus. But I started my graduate degree December 2020. Like pandemic. They were like, no, sorry, you can't be on campus unless you're like essential. I was essential because I was working in a lab that had a lot of research plants in the greenhouse that needed to be taken care of every day. And so I got to go into my office and I could maintain this aquarium. But I ultimately ended up bringing it home. And so then I was trying different compositions of like going from like light green to yellow to orange and red and just trying different plant species. And then I've tried Wallstad method. So this is just a bowl that I had sitting on my desk. Just soil at the bottom, sand capped on top, and just shoved a bunch of plants in to see if it would grow. And yeah, like 3 years later, that's what it looked like. But there was, so going into, like starting this hobby and learning more about it, I was on forums like every single day. I'm like multiple hours a day, like plantank.net, Aquatic Plant Central, like looking at all these forums, but there was just certain things I wanted to know about aquatic plants that I couldn't get information from the hobby. I needed to like basically go back to school. I wanted to learn like how to identify plants, how to like formally name them if you find a new species, like learn about the genetics of plants. And so, living in Minnesota at the time, I got to see aquatic plants all the time. There's lakes everywhere. And so sometimes I'd run into plants, I have no idea what they are. And I didn't have an app on my phone at the time that could just tell me what it was. And so this isn't an example of Minnesota, but this might be something somebody's familiar with. Let's say you go to a lake and you see these two plants growing. This one's underwater and this one's at the surface. What do you think you see here? Does anybody know what this plant is? It's not hibiscus. Does anybody have an idea? No?
Water pass.
Pondidaria azurea, or what was formerly known as Icornia azurea.
I was going to say that.
It's another federally listed noxious weed.
We believe you're.
If you have this in your aquarium, like, you might be on a list. I don't know. It's a really pretty plant, but it shows that, like, if you see it growing completely underwater, how much different it looks like than when it's growing at the surface. They look like two totally different species. They don't even look like they're related at all. Oh, they're both the same plant, yeah. And so, like, here's a different situation. This is just one plant. I know this is all from the same plant, but all of the leaves have a slightly different coloration pattern to them. And so my question is, like, why do we see these types of things happening to plants? Why do they look different where they're growing?
Available nutrients.
Yeah, that's a factor towards something that's called phenotypic plasticity. And so it's a plant's ability to change the way that it looks depending on available nutrients, light, water temperature, water parameters. So like some people have like these jungle valves. Like these are huge. This is like jungle val, right? So I bought a jungle val from Dustin's fish tanks, and I grew it in the greenhouse, and I was able to grow it like this tall. Like jungle val that's like 10 feet long, right? I just tortured it. I put it in like super crappy topsoil clay and put it in a bucket in the greenhouse and just let it sit there for like 6 months. And it grew leaves that were only like that long. It was pathetic looking. But it's the same plant that grows like that big, right? So what we're seeing here is phenotypic plasticity, and it's actually one thing that Vallisneria are very good at doing, as you can see. And so a little bit of background about vals. They're found worldwide. You can find them everywhere around the world. And so here's a map that I pulled from GBIF. And GBIF pulls together information from herbarium specimens. So if you go to an herbarium, you get the dried plants that are pressed and glued on the paper. It gives the information of where they were collected, who collected them. And then this map gives occurrences from iNaturalist. Who uses iNaturalist here? Some people? Yeah, it's a great, great app. You can document biodiversity on your phone, and you can take pictures of plants and animals, and you can post it online with what you think is an identification for a species. And if multiple people agree with you on that species, it will become research grade, and then it gets shot onto this map. And so what we know is currently there's about 14 species that are accepted. There are 14 species that are accepted. So within taxonomy, there's a term called accepted species or not accepted. Accepted means like that is the name that's currently correct. So there's 14 listed. But I disagree with this number, and a lot of other researchers that are in this field also disagree with that number.
You think there's more?
I do think that there's more. So Vallisneria monocots, meaning that they are related to grasses and sedges. And their generalized growth habit is they grow from a rosette or at the crown base, and then they grow their leaves straight up from there. And they reproduce asexually by making runners, or what's called stoloniferous growth. They make just genetic clones of themselves in the runners. But again, with biology, there's always some exceptions. In Australia, there's 3 species of vals that grow along a stem, like a regular stem plant, rather than just at that crown. There's Vallisneria caulescens, Vallisneria triptera, and Vallisneria rubra.
Are any of those in the hobby?
Yes, so I was able to import Vallisneria triptera. This got sent with it, but it ended up dying because it's so delicate, and it took 6 days to get here from Australia. So I was able to get it. If you look on Facebook Marketplace on like quick auctions, there are a few people who still will grow this. I know Tom Barr had a huge like 100-gallon aquarium just filled with it because I sent it to him. He grew the crap out of that plant. Way better than I could. I got it to look really nice in a pond, but growing it inside in my aquarium, it was really difficult. It requires really hot water, like 86 degrees Fahrenheit or higher. Yeah, like the hotter, like basically bath water.
It requires crocodile food.
Yeah, salties. Yeah.
So in my 40-degree water, it's not going to do that.
No, it's going to be really unhappy. So then what's cool about Vallisneria is that they are also dioecious, meaning that an individual plant is either male or female. Yeah, so this plant on the left is a male, and it's producing these spades. So these spades right here, they're basically hermetically sealed balloons that contain hundreds and hundreds of tiny little male flowers. And so once those spades mature, they actually crack open at the top, and the male flowers start to get released like one at a time. And they look like these little yellow dots that will float up, and fish love to eat them. And then on the left here are some female flowers. I'm sure people have probably seen. These are like more commonly seen, the female flowers that rest at the surface of the water. And so the female plants will produce these on this long stalk. It's kind of like stretchy. It's called a peduncle. And it's one of the reasons why more female plants are collected than male plants, because they're more easily observable in the wild because you see those female flowers resting at the surface.
Are all Vallisneria plants like that, the male and female species?
Yes.
Okay.
So all vals have either, like an individual plant will either have male flowers or it will have female flowers. It will not have a mixture of both.
Now,
And even with the clonal runners, it won't produce different sexes.
so right now I have the yellow thing.
Yellow dots?
Yeah, so
Yes.
I have a single parent plant, so all of my plants are males, all the runners I bring, they're
Yep.
all males.
Yeah.
Because I've never had one flower, and I always wondered why.
Yeah. So
I collected it in 2020, and I've never had a flower, and I've had Val's flower all the time.
if you see little yellow dots
Yeah,
on the surface of your water, I'll show a picture of what those things look like
I
up
have them
close.
now.
So typically we see vals make a solitary female flower. That is, on that one stalk, they produce one female flower. But in Florida and parts of Texas, a researcher, this Rohal et al. 2021, saw that there was some plants that were producing an umbellate of female flowers, which is kind of odd. And these plants are entirely pollinated through wind and water movement alone. They don't facilitate animals. They don't facilitate like any insects or like beetles or hummingbirds or nothing. It's all based on wind and water movement. So there you can see the tiny little male flowers surrounding a female flower on the surface of the water. And so once those male flowers come into contact and they pollinate that female flower, the flower will contract and coil, the peduncle will contract and coil down. Yep, if you've seen that in the wild, it's the reason why the type species, when Linnaeus saw it in 1753, he named it Vallisneria spiralis. He didn't name it spiralis because the leaves spiraled. He named it spiralis because that female flower spirals And although these flowers are really tiny, they do produce different coloration and patterning to them. And so here's some that I had in my collection that flowers. I have Vallisneria australis, Vallisneria spiralis, Vallisneria gracilis, and Vallisneria nana. And I brought the nana with me today to donate for the auction. Yeah?
What's the evolutionary significance of producing a colored flower if you're not, like...
That's a good question. I literally can't answer that. I have no idea. So one thing that I was guessing was that flower, like I had guessed this like during my project, was that flower color is directly correlated to where they grow. So obviously the red pigment that's being produced in these flowers here, so Vallisneria nana and Vallisneria gracilis grow in the tropics of Queensland and Northern Territory, where there's sort of hot tropical sunlight, and so they're producing this red pigment, which is called anthocyanin. Whereas these come from temperate locations and they have white pigment. They don't need that. The anthocyanin is like a UV protectant for plants. So here's the male flowers that I took pictures of under the microscope. This left picture, this is the spathe, that male spathe that holds the hundreds of male flowers. I pulled it open physically. I didn't wait for it to crack open, and I pulled them out. They look like little spaceships or like little balloons. And yeah, when they float to the surface of the water, that's what they look like. But then once they hit the surface, they actually crack open, like they crack open, and the sepals, the three sepals, act as like a little flotation device for the flower to float around. Because on this specific male flower, they have two anthers and then a cluster of pollen grains sitting on top of them, and they don't want those in the water. They want them to be up so they can come in contact with the flower. And so those three sepals crack open and they act like a little boat.
So they're not attached to the male once they leave that?
No, that male spathe is not the flower. These are the flowers.
Right, that just opens and it's not attached.
And so that balloon-like structure holds hundreds of these, and you can see how small they are. That's the tip of a dissecting pin. And so these things are about 1 to 2 millimeters tall. And they're so small that under a higher magnification, you can count how many pollen grains are on each flower. I wasn't going to. That was not going to be a part of my project because that would have been a pain.
But you did anyway.
And I did not do it. I didn't. I was so tempted, but timing it is really tough because sometimes I'd go to the greenhouse like at night and I'd just go to like refill the giant tub of water, and I'd be like, oh my god, they all flowered. Like, what am I going to do? Am I going to do it right now at like 6 o'clock in the afternoon? So I also wanted to highlight that there's some really good leaf color patterning that's different in like all of these Vallisneria species. So you can, it's kind of tough on this TV. Maybe if you come up later, I can show you a really good picture on my computer what they look like. They have different coloration patterns. And then this picture on the right shows one of the things that's unique to Vallisneria. Well, it's not unique to Vallisneria. It's a feature of Vallisneria, which is the serrated leaf edges. Like they kind of have sawtooth blades. And one of the features that was identified before was counting how many serrations there are per millimeter. Yeah, that was kind of annoying. Yeah. Speaking of previous research, in 1982, a paper came out from Richard Loden. So he wanted to assess all species diversity around the world. And at the time, oh, thank you for the lights, appreciate it. And at the time, there was about 9 accepted species. His analysis, he said, looking at leaf structure only, it's no good. We're not going to do that. We're going to look at just the floral traits because we think that based on phenotypic plasticity of the leaf sizes, it's not a good characteristic to be able to determine species. Instead, he's going to look at flowers because he felt like flowers were more stable and that depending on the environment, the flowers still stayed the same. And after his study, he decided there's only 2 species of Val, Americana and Spiralis. That's it. And then everything else is just a variant or a subspecies. In 2008, they wholly disagreed with that. Les et al. Came out and they did genetic work.
Yeah.
They looked at both molecular and morphological data to assess all available species that they could gather around the world. And they estimated the group's first phylogenetic relationships. So they used parsimony and Bayesian analyses, and this might go over people's heads. It's just 2 ways to look at different ways to assess the tree of life of this group. And they recognized a much more speciose genus than Loden. So with that background information at hand, I'll go over the direct, like the absolute of what I did with my project. And then also what led to getting my first publication. So my research objectives got narrowed down a lot. I came into the meeting with my advisors with like a ton of stuff I wanted to do. I wanted to look at like pollen grain information, like leaf structure, and they're like, no, just focus on a few things. So I wanted to estimate phylogenies using maximum likelihood. So maximum likelihood is a different method to create a tree of life using a different analysis method. And I wanted to see how it differed. So first I wanted to see, like, does it produce any different results than what they got in 2008? That was the first question. Like, if I use maximum likelihood, which is right now known to be better at resolving relationships when you use a ton of DNA data. So I'm talking like hundreds and hundreds of thousands of base pairs for each like section of DNA for these plants to analyze how they're related to one another. So I kind of want to lay down the groundwork. Like, is this a good analysis type to use for later methods? So I also wanted to expand on the work from Les et al. 2008. So I got some plants that they didn't get to use in their analysis, which is pretty cool. I wanted to use the phylogeny that I got and morphological data to assess taxonomy. So I wanted to determine how many species do I think are in Vallisneria. And then I also wanted to use the molecular data to help identify cultivated plants, so like the stuff we grow in the hobby, and the ones that I collected out in the wild. So in order to reach these goals, I wanted to increase taxon sampling. That is to say, I wanted to get new plants that hadn't been researched before, and I wanted to capture their DNA and get them sequenced. It was difficult to obtain plants because I was doing my project during the peak pandemic, and so I wasn't able to make a trip out to China to get plants. Wuhan, where like a couple species were described. Getting to Australia was going to be difficult, so yeah, I sourced some from the aquarium trade, as in I just bought them from hobbyists. I bought them from different pet stores. So here's a list of the ones that I was able to determine what the species were. But there's and there's this one that I got from Joe Harvey. I'm like a fanboy of Joe Harvey because of his Dutch aquascapes. This one right here, the red arrow pointing to the left, he used it in his aquascape. It's called Vallisneria leopard. Has anybody seen that one?
I've seen it, but isn't it a cultivar?
We'll go over that in a second.
Okay.
So I knew I was gonna get flack for this from reviewers when I wanted to publish this data. If you're getting plants from hobbyists, how do you know where they came from? How do you know if it's the right species name? And that is a problem because you have what I determine these plants are, and what they were sold to me as. So I bought Jungle Val from Water's Edge. It was in Lawrence, Kansas. There was a pond supply store. They also sold Italian Val and Corkscrew Val. But like with common names, sometimes you don't know what they are. So I was able to go out and actually do some field work. I went to near my house where I grew up in Minnesota, and then I was also able to make a trip to Florida to collect plants. In Minnesota, I collected from this lake called Square Lake. It's known to be very clear water, like visibility up to like 40-something feet. Super clear. The lake gets to like 86 feet deep, and you can find plants growing all the way down to like 42 feet. Like if you want to collect plants down that far, you need scuba. And the lake is actually really well known for scuba diving. And so this is just north. This is about 20 miles north of the Twin Cities, and you can actually see the St. Croix River here. This is the border to Wisconsin. Wisconsin. Yeah. So while the lake, I said, had plants growing down to like 40-something feet, I limited myself because I was just free diving down to like about 10 or 11 feet to collect plants. Mostly it was along the shoreline, just hanging off the side of my cousin's boat as he like trawled around with the electric motor. And I literally just had my whole body in the water, just like, stop, right here. I just ran down and grabbed plants.
Was there any indication that there may have been something different growing on the bottom versus near the surface?
Yeah, so this lake specifically this was like almost predominantly dominated by Chara. So it's a macroalgae. And so like down at the bottom of the lake, there's like a good 6 to 8 inches of mat of nothing but chara. And like the valves at the bottom of the lake, you could see them just poking up through that mat. And so what happened, I can give a quick story about this. Square Lake used to be very heavily regulated on like, the surrounding homes couldn't use fertilizer in their lawn, and no motorized boats were allowed on the lake. And then they started to relax those regulations. The lake got huge algae blooms and became eutrophic, and so now a ton of the aquatic plants started growing because all of the fertilizer and nutrients started getting mixed up in the lake. And now they've tightened those regulations again. The lake is clear again, so I was lucky when I went there. The visibility was like 40 feet again, which was great. But the damage is done, right? So now there's that huge mat of chara when typically you could actually see sandy bottom. So I transported those plants back to Kansas when I went, back home. And I also was able to go to Florida, in Central Florida, to collect aquatic plants as well. So I originally made the idea to go to these sites just as a trip by myself. And my wife was like, "Hey, our 10-year wedding anniversary is coming up. Why don't we make it a dual-purpose trip?" I'm not going to argue with her, so I said yes. Like, yeah, let's do it. Okay. So we got an Airbnb on Lake Hernando, and it was really cool because there was Vallisneria growing right off the dock. It was awesome. I was like, this is amazing. If you ever stay there, yeah, there's really good Airbnbs on Lake Hernando. But I was originally there because I saw this site on iNaturalist. Somebody took a picture at this site. This is Little Jones Creek. It's a spring-fed creek that's just north of Lake Panasoffkee. It's in between the Gulf Coast and Orlando. And you can see that the valves here are pretty dominant in the water. This water stays about 72 degrees Fahrenheit all year because it's spring-fed. It's a freshwater spring. And these valves were absolutely the biggest Vallisneria I've ever seen in my life. 10 feet long, easy. Yeah, like when I went there, you saw the gallon sandwich bag that I had, or the gallon bag I had for the valves in Minnesota, yeah. There's no way I was going to be able to get these in the bag. I had to like go back in the water and try to get the edge ones that were like the small runners, because these things were like easily 2 inches wide and 10 feet long. They were huge, yeah. But there was one day where I was like,
Wallstead had that.
Wallstead that, yeah. And then get the picture where the tank's over there and I'm holding onto the lead over here, yeah. But one of the things that we did, one day it was just like we had an open day. And my wife's like, let's do the kayaking tour on the Crystal River. So it's a freshwater spring that feeds into the Gulf in Kings Bay. And so when we got there at the dock to like get on our clear kayaks to maybe possibly see manatees, I saw all of these valves in the bay, and this is actually a brackish water, so it's half salt, half fresh. And so these Vallisneria covered like the entire bay. Like you could not see the bottom. It was nothing but Vallisneria. There were so many.
Is that all Americana?
That's a good question. Because when I saw this was already after I had collected the ones from Lake Hernando and from Little Jones, and I was like, these don't look anything like the ones that I collected from that creek and that lake.
But they're growing in a different habitat.
That's the question. Do they look different because they're growing in half salt water? I don't know. So I grabbed some and brought it home. Got a permit afterwards, retroactively, let them know I grabbed some plants. Everything that I did to collect plants had to be on the up and up because I was getting this work published and they require they check. They're like, send us information about the permits you got. Like, okay, I got it. Yeah. Well, you saw how many were there, right? I took 3 home. Like, there was thousands on the shoreline from them floating up and just like hitting the shore and drying out. So I just really walked up and grabbed a couple and put them in a bag, brought them home. But I also got plants sent to me from Australia, from who I now call my friend Dave Wilson, who is like the most Australian person I've ever seen in my life. Like, I've talked to him on Facebook, like the video chat. His accent is so thick, it's like so Australian, and I love it. It makes me smile every time I talk to him. He's a really good guy. If anybody makes their way up to Darwin, Northern Territory, you have to look him up. He owns a small business called Aquagreen, and he focuses only on growing native Australian stuff. So native plants, native fish, native shrimp. He'll take you out on his boat, and you can go fishing for Jardini arowanas, which are native there. They call them Saratogas. And when he catches them, they're like that long. They're huge. Yeah. Yeah. What?
What do they taste like?
I don't know. I think he mainly catches them to grow for his aquaculture. So the plants he sent me 5 different types. He sent me 6, but one ended up not making it. So the ones that did make it, you can see that there's 4 collection points in Northern Territory, and then this one all the way down here in Queensland. And the one in Queensland, that's the one I brought today. There should be 5 bags of that val. And so through Tom Barr, I messaged Tom Barr one day, and I was like, Tom, I'm working on a master's project. I need help. I need plants sent to me from around the world. Who can I get in touch with? And he's like, he listed off some names. One of the names was Dave Wilson. So I got in contact with Dave. I emailed him, and he's like, I've tried. He's like, yeah, I'll absolutely help you. I have some plants that were supposed to be in that 2008 paper. They never made it in. So I want them to be researched. And he's like, I got bad news though. I've tried sending plants out of the country twice, and I've failed both times. The Australian customs flags them immediately, destroys them. I'm like, that's not good news, Dave. Like, I got 2 years to do this project, right? And I need plants like right now. So what can we do? And he's like, I don't know, look online. So I looked online. Australian government gives these 9, and I'm going to put in quotes, easy steps to export plants out of the country. It's just 9 steps, right? No. Each one's got this little drop-down bar, and you hit that, and it just expands out to like 10 more pages. And you're like, ugh. And of course, right away, check what type of goods you're exporting. All right, I'm exporting aquatic plants. Okay, so the drop-down is, are they prohibited, or are they not prohibited? Okay, and I said, oh, how do I figure that out? Well, here's a list of everything that we have that's prohibited, not prohibited. Vallisneria was not on that list. So what do? Contacted them again, and they're like, no, you're lying. You didn't see it. I'm like, no, it's there. Vallisneria is not on the list for either prohibited or not prohibited. Like, a week later goes by, they say, all right, we've determined that Vallisneria is indeed not on the list. We'll get back to you. So then, like 2 or 3 weeks later, they get back to me and they said, all right, we formed a committee. We formed a committee, and we've determined that, Vallisneria are not prohibited, so you can move forward. And during this whole time, Dave's emailing me back, like, are you ready to just smuggle these plants in yet? Like, is this annoying? This is annoying, isn't it? I'm like, yeah, but we have to. I'm like, we have to be persistent. We have to do this the right way. And so then, check importing country requirements. This was super easy. You make a profile on USDA website. They send you the documents. You fill them out, and then they say yes or no. You just let them know what you're bringing into the country, and they just okay it. It literally took like 3 days. So I had to register Dave as a verified exporter. So I had to get him a license. He didn't want to do it. I'm like, I'll do it. I don't care. I'll do everything. So I got him listed as an official exporter. But then you have to fill out all this electronic document paperwork. They won't email you stuff like USDA. You have to hire a company that gets the documents. Then that company gives you the documents. And you have to pay that company to do that. So unnecessary. And so that's why I say, I understand why people smuggle plants into the country with their plants, but I don't advocate for it. But I understand why, right? I get it.
Everyone hates bureaucracy.
Exactly. Dave's just like, I could just send you a box of clothes. I'm like, no, I can't do that. So we made it through all these steps. It took 5 months to get through this whole thing. And yeah. Clock's ticking. I'm working on my project, right? So once the plants got sent to me and all the ones that I collected, I had this, it's a common garden. I call it a pond. My wife's like, is it technically a pond? I'm like, it's a 50-gallon Rubbermaid tub in the greenhouse. It's not really an aquarium, right? Would you call it a pond? It's like a patio pond, but this was in the greenhouse at the university. And if there's any hardware people that are interested in what I used, I had it hooked up to an FX4. And I did use CO2 injection because I wanted these plants to grow really fast. And so I made a Sergey's reactor, highly recommend if you have a big aquarium to use a reactor like this. It is very efficient, and it's actually pretty cheap. You can make them for like 20, 25 bucks. And so after I had the plants growing in the pond all together in a common garden environment, I waited about 6 months before I started to measure certain aspects of the plant. I wanted to look at like leaf size, leaf width, how many serrations per millimeter there were on the leaf tips. So I had to wait about 6 months to do that. And so like I said, one of the vegetative features that I looked at were the leaf tip serrations. So this is the one that I collected in Florida. You can see it's a much bigger plant. And you can see like just there might or might not be some variation of how many serrations there were per millimeter. I determined that it wasn't a useful character, so I ended up abandoning that later. But then I also was able to look at different floral characteristics when they became available. So when these plants started flowering, which took a while, I was able to pull them apart, look at them under a microscope. So, you know, like typically with flowers, the really showy part, the colorful part is the petals. Well, the petals on these things are that little white dot that's above that laser pointer, this thing right here. That is the petal. They are like a 0.5 millimeter tall and like a 1/4 millimeter wide. They're so small.
So what's the purple?
The colorful stuff is the stigmatic surface. This is what the pollen will attach to fertilize the plant. And so then I also went to the herbarium and they let me use their fancy microscope with a camera to take a picture of the male flowers because I wanted to see just how ridiculously small those were.
You weren't
Yes.
worried about
Did you dye that all they just green by
No,
itself?
it's just because of the image, it was just it turned up yellow. These are clear. They're like no color at all to them except for the pollen is yellow.
So like you didn't dye it.
I didn't dye it at all, no.
It's just how it showed up.
Just on the picture, yeah. So I was able to use those features that I was measuring, yeah.
You're not worried about hybridization?
So a caveat to how I was doing all this work was anytime these produced flowers, male or female, I got rid of them. So anytime I saw female flowers, I just chopped them off. I didn't allow anything to go to seed. Yep.
If they're only like this big, how do you see them?
Because they float on the surface of the water. So when I'd go into the greenhouse and I'd look at the pond, you'd see these little yellow dots everywhere over the top of the water, and I would just take a little glass vial and I scooped them up, and I could see them like, okay, those male flowers, and I bring them to the lab.
Do you have any issues, for example, like collecting only male or only female plants?
Yeah, and so that's reflective on this. So good to have numbers in this matrix, bad to have dashes. The dashes are because I couldn't guarantee that I was getting male and female from one particular species. So like when Dave sent me plants from Australia, some of them were all female. Some of them were all male. We couldn't guarantee because when you collect them, sometimes you're collecting them, they don't have flowers on them. So I made this just because it was useful to show that I actually did work in my master's, and this was a lot of work, but it ended up not working out very much because it was so difficult to get both. And because some of the characters were showing multiple states. So like, this could be, max leaf width is between a certain range, and then the next jump is between this certain range. It's like, well, when I measured max leaf width, they fell within the ranges that were pre-established. So it got 2 scores. Well, that's not helpful to me. I wanted to have 1 score, and for it to have an X means that I just couldn't either measure it or it didn't match up with anything. So this ended up, I abandoned this, especially for the publication part. They were like, if it didn't do anything, then why write about it? And so to do DNA extractions, I dried plant tissue on silica, and then I put them in these epi tubes with steel beads, and then put them in this little archaic device right here, which is a reciprocal saw, and it shook them back and forth. So I froze them in liquid nitrogen, shook them back and forth to break up that plant tissue into a really fine powder. The idea is to break apart the plant cells to make DNA available for me to get out. they make a really fancy piece of equipment called a geno grinder. It's like $20,000, but the one professor I was working for was like, I'm not spending $20,000 on a piece of equipment I can make from Home Depot for like $200. So he literally went to Home Depot, bought this reciprocal saw, brought some 2x4s and some C-clamps, and just nailed this thing down to the table. And so he made the press here, so like you take all the epi tubes and you put them in like a little holder and press the wood together, and then it just shakes it back and forth really fast to mix those beads up. And it made the, like, I would pre-warn like all the labs around me, like I'm about to use the, we called it the de-annihilator. I didn't coin that term, another grad student did. I won't take, it's super loud. It's like, because it's a reciprocal saw, right? And it's got this, like all this metal, it's attached to a table, it made a ton of noise. But it did the job. It didn't need the geno grinder.
How's your hearing?
Hearing's good. I had headphones in. So then after I got the DNA out using those kits, I verified that I had good DNA with a nanodrop spectrophotometer and a qubit fluorometer. Qubit fluorometer and a nanodrop spectrophotometer. And then it produced, like this nanodrop produces this graph to show me that I have good DNA based on, it shines a light through it. It says, yep, you have DNA in this quantity. It's good. You don't really have anything else that's garbage. So after I did that, I used that DNA and I ordered PCR primers. And so what primers do is you have like this whole genome on the plant, all of the A's, T's, G's, and C's that make up the plant's DNA. And I wanted to target 3 specific regions. And so these primers, they adhere to the forward and back side of that, and then the PCR will amplify that region like millions and millions of times. And then that amplified product, I had to verify using...
Define amplify for the scientist here.
So initially what the PCR does is it will take the DNA and it will adhere the forward and reverse primer to both sides of that DNA chunk that I want to look at, and it will copy that.
Yeah.
And every time it goes through a cycle, it will double the amount of copies that I just had. And so it does that over and over and over. So every single time it's doubling, 2, 4, 8, like it just keeps going over and over and over until I get like hundreds and thousands or millions of copies.
I'm very familiar with selling, but
so to do the work, you don't just want like one copy of it. You want a lot of copies so that you have like a good backstep, like a quality control, really. So then I verified that I had the region that I was looking for based on the amount of base pairs that I was getting in that amplified product by using something called gel electrophoresis. And so this bottom is a ladder, and this ladder is known for the amount of base pairs that are in DNA. And so I could verify that I had, around, for this one, I think it was around 666 base pairs was what I was looking for. And so I knew that this one right here was about 700 base pairs was the ladder. But it's really cool because there's these 3 that were a little bit lower than everything else. So I was like, was it bad amplification? No, what it turned out was, this is Vallisneria spiralis. This was one of them that I got that I thought was spiralis, and here's another one that I thought was spiralis. So it kind of shows that they might be closely related based on the fact that they are matching up those 3. So then after I did this, I verified that all the DNA I grabbed was good, all the amplifications I did was good. I sent everything into a company that would give me back the actual DNA sequence itself. And so they sent it to me just in a text format. It's literally just A's, T's, G's, and C's in a text line. But that doesn't really help me out because sometimes they might not be reliable. So what they sent me as well is these chromatogram data. And so how they sequence DNA is they literally go down the line starting at DNA number, or at nucleotide number 1, they shine a light through it. And then that light will produce a peak and tell them what nucleotide it is, whether it's an A, T, G, or C. And so they do that for the entire thing. They have a machine process that does it really quick. I didn't have that, so I couldn't do it myself.
Nobody does it themselves.
So I was able, I hand checked, because I'm not working with a ton of base pairs, I was able to hand check this myself to verify that everything they were sending me looked good. You want these nice peaks. Sometimes it gets messy, it's like wavy, it doesn't really look good. So I usually would just cut off the section there and say like everything after this point is not reliable. And so what you hope to do is you sequence forward and backwards so that you can get those sections that are a little bit bad to maybe overlap so that they look good in one direction or the other. And then I assembled all the DNA using a program called CView. And I also pulled gene sequences from GenBank. So the stuff that was used in Les et al. 2008, they accessioned all their gene sequences in GenBank. So I can just pull off all that information and use it in my study as well. And then I also added the sequences that I was able to generate to GenBank as well. And so here are the 18 plants that I used in the study that were just collected by me. There was 51 others that I used from the previous project. But the 3 that I collected in Minnesota, I collected from all around the different spots of the lake. With the 3 gene regions that I used, they were all identical. So I can't for certain say that the plants that I collected are all exactly the same, just clonal individuals of one another. All I know is that the sequences that I collected from them were all the same. So for everything else that I did in the study, I only picked one of them to do analysis on because 3 identical is not going to do anything for me. For building the tree of life or building the phylogenies, I used a web software called IQTree. It does phylogenetic analyses using maximum likelihood, uses Model Finder, and I ran the analysis using 100 bootstrap replicates for node support and an approximate Bayes test for branch support. And I also ran an ancestral state reconstruction in R to estimate the ancestral state of Vallisneria. So remember when I said there's those 3 species that grow along a stem and like everything else grows from a base crown? Well, this program will go through the phylogeny. It'll basically say like, oh, we think that the earliest diverging ancestor had a caulescent habit or it had a rosette habit. And so this is going to be able to tell me what they thought was changing throughout the tree of life, if there were changes at all. And then finally, I wanted to know, where did Vallisneria originate from? So where did they first start popping up in the world, and when did they? I was fortunate enough because somebody already did this to the whole family, Hydrocerataceae, which Vallisneria belongs. So I was able to use the information that they had and apply that to the tree of life for the phylogeny that I was producing. So what did I find out? Using just the one gene sequence from the nuclear region in these plants, I was able to produce a phylogeny here. So this is a tree resulting from just the ITS data. And then all of the plants that I collected personally or got sent to me from Dave, or the ones that I purchased, are highlighted in yellow or in orange. And then everything else is the ones that were used in previous studies. So here's the tree that I was able to produce from just using the chloroplast DNA. And then here's the one that's produced using the combined. So using all 3 gene sequences, I was able to produce a phylogeny using those. The one in the publication looks a little bit different. I'll show that later. And that's just because the reviewers had a big issue with me using hybrids in, spoiler alert, hybrids in this phylogeny. So I said I wanted to learn what did the evolution look like for plants in Vallisneria that had this caulescent habit, or they had stem growth, or the ones that we typically think of vals, which is the rosette growth. And so this analysis determined that the first ancestor within Vallisneria most likely had a rosulate habit. And I say most likely because it's about 73% probable. And then moving up the tree, this ancestor was about equally likely to have either or. And that's like not great. That's why I didn't make it in the publication either. But we know that within this lineage, the ancestor definitely had a rosette habit. That's because all the plants that are encompassing this part all have rosette growth. And yeah, the troublesome group was this group right here that I have highlighted in pink. And so this is the tree that was developed by Les et al. In 2008, and this is the tree that I developed for my project. And so they showed that the three coalescent groups were closely related to one another, but they had really low support. That 32, that's the bootstrap replicate support, that is a really low number. You say that's very low support. And that's because some of the times that they produced the tree, it looked like this one. And that's the one I also have low support, because sometimes when I produced the tree, it looked like that one. This analysis had trouble placing Vallisneria coalescens. It was kind of like an outlier. It didn't know where to place it, whether it was closely related to the other two, or whether it was the ancestor to the entire group. It didn't know. So that's why it was tough to infer evolutionary changes with that growth habit in this group. But this one was a little bit nicer. This was like, where did Vallis come from? Well, the analysis shows that Vallisneria come from about Southeast Asia. That's where they originated. They had a dispersal event where they went from Southeast Asia over to Europe and down into Africa. I'm sure there's got to be at least one person here who has a rift lake, like Lake Malawi Val, right? Anybody? The last club, there was like 3 people who had it. No? Okay. There, in the Rift Lake, there's a Vallisneria that they thought was a different species. That Les et al. 2008 paper determined it was just Vallisneria spiralis. It looks different because it's growing in that hard water in the Rift Lakes. There was a dispersal event from Southeast Asia into New Guinea and Australia and New Zealand. And then here's the really odd one. Somehow, Vallisneria made their way out of the water and across the sea to North America. I can't tell you how it happened. I don't know how. I've consulted with ornithologists. Are there birds that go from Australia to North America? And they're like, yeah, there are actually seabirds that go from Northern Territory and they land somewhere around Alaska and the west coast of Canada. So it is possible that either the fruits themselves were digested and then they pooped them out in America, or the seeds were collected on their feet in mud, and then as they flew over and they landed, they got deposited there. Because in every analysis type we see that it goes from Australia into America. And it was really cool because it went from Australia back to Asia. So the ones that we find in Japan, the Vallisneria asiatica, the mini twister, the really, really tightly spiraled ones, that's the newest diverging species that we see, is the one that's found in Japan and other parts of Asia as well. So the question is that I wanted to answer, how many species do I think there are? I'll focus in on this group right here. This is from North America. So right now, official taxonomy, all the officials that are part of like Kew Gardens and international plant naming systems, they say that there's one species found in all of North America. It's called Vallisneria americana.
What about neotropics?
That's a good question, because I have Neotropicalis up there. I don't have it listed as americana. So right now, Neotropicalis is synonymized with americana, because if you think that there's only one species between these two, the oldest name wins out, and the older name is americana.
Right.
Yeah. But my study and the studies that have been done previously from Don Less in 2008, we keep finding the same thing, that when you collect plants from the subtropics of Florida and in the temperate zones like Minnesota, every time you add them to this little tree of life, or this phylogeny, they always split up into 2 groups. The neotropical ones go together, and the temperate ones go together. And one of the things that was argued was, okay, well maybe the genetics might be showing something, but what about the way that they look? We think that the only reason that the neotropical ones in Florida look different is because they're growing in the tropics. They get to grow all year. The ones in Minnesota have to overwinter. They die off. They don't die off. They winterize, and then they pop back up in the spring. So it's just the growing season that's causing them to change different. This is 2 years later of them growing in the same pond. So this is Vallisneria americana that I collected in Square Lake in Minnesota. And this is Vallisneria neotropicalis that I collected in Florida. And as you can see there, they look quite different from one another. I had been growing them in the same pond for 2 years.
Question?
Yeah.
So is there any possibility, I don't know how plants work well, but that if they started in one setting, they're just kind of programmed in to grow that way even if you change the setting, versus if you went from seed with those two different populations in a different habitat?
The one of them is from seed.
Okay,
Yeah.
there you go.
So the phenotypic plasticity, because they produce clonal runners, like obviously if I collect those